Good morning, students. Today we will be trying to learn about the mechanism of the cell injury. So, whenever there is an injury to a cell, there are different types of injuries what the cell can have. So, when we try to enumerate the list of causes of the cell injury, we have physical causes, chemical causes, microbial causes, nutritional causes, genetic causes, immunologic causes, and others. So whenever an injury occurs to a cell, what would be the mechanism? How the cell will try to suffer from various types of injuries. When we try to further classify the types of cell injury, it could be a reversible type of cell injury or an irreversible type of cell injury. So the principles that are relevant to the most forms of the cell injury are the cellular response to the injurious stimuli, the consequences of the cell injury, the biochemical mechanisms acting on several essential cellular components, and any cellular injury, the stimuli may simultaneously trigger multiple interconnected mechanisms to damage the cells. When we look into the biochemical mechanisms, the various steps would be the depletion of the ATP, there would be a mitochondrial damage, and because of this mitochondrial damage and further the consequences would be the influx of the calcium and the loss of calcium homeostasis. There could be accumulation of oxygen derived free radicals. When we say oxygen derived free radicals, we have few of them, that is superoxide anion, which is designated as O2 minus, and we have OH, which is called as hydroxyl ions, and then we have H2O2, which is called as hydrogen peroxide. So these are all the oxygen derived free radicals which may be accumulated during the process. And apart from this, there can be a damage to the membrane and damage to the DNA and protein as well. And whenever there is a depletion of ATP, there can be a hypoxic or the chemical type of injury. And the ATP generally is produced in two pathways. One is the oxidative phosphorylation and second is the glycolytic pathway. And when you look into the diagram, this is the overview of the cellular respiration. Here you can try to see the glucose, it is trying to get converted into pyruvate and then further into acetyl coenzyme A, and then it tries to further involve with the citric acid cycle. And when you see that these reactions they occur along with four steps. One is the glycolysis, second is the pyruvate processing, and third is the steps involved in the citric acid cycle, and fourth would be the electron transport and chemiosmosis. And this is again another picture displaying the oxidative phosphorylation, wherein you see the various parts. This is the outer membrane and this is the cellular matrix which is within, and here you see the inner membrane, and then when you come outside, this is called an intermembrane. So these are all the various parts of the cell where the oxidative phosphorylation takes place. Now, when we look into the major causes of the ATP depletion, they are the reduced supply of the oxygen and the nutrients, mitochondrial damage, and actions of some toxins. Now, when we look into the requirement and the functions of the ATP, why is the ATP required? It's mainly required for the membrane transport, for the synthesis of the proteins, lipogenesis, deacylation, reacylation reactions necessary, phospholipid turnover. Then what would be the effects on the critical cellular systems? There could be a reduced activity of the sodium, potassium, ATPase pump. There could be altered cellular energy metabolism, failure of the calcium pump, and reduction in the protein synthesis, and there can be an unfolding of the protein response, and there can be a further damage to the mitochondria. And here when you look into this picture, so whenever there is a kind of an ischemia, so due to the lack of blood supply, sometimes the cells suffer from this kind of a stress. So whenever there is an ischemia or the hypoxia, naturally there is some kind of stress on the this particular organelle of the cell, most important organelle of the cell, which is called as mitochondria, wherein you see that there is a decreased oxidative phosphorylation. So whenever there is a decreased oxidative phosphorylation, you see various kinds of changes that they occur in the cell. Now let us try to know further. Because of decreased oxidative phosphorylation, there is decreased synthesis of ATP. Now because of the decreased synthesis of ATP, you see that the sodium pump functions are altered and there is an influx of calcium, water and sodium, and there is efflux of potassium. And further, when you try to look into the morphological changes what occur in the cell, there can be a swelling of the endoplasmic reticulum, and there could be a overall cellular swelling, and there could be loss of microvilli bleps. So these are all the changes which occur on one side. And when you look into the other aspects, due to decreased ATP synthesis, there can be increased anaerobic glycolysis. And because of this glycolysis, naturally there is decreased glycogen content present, and then there can be an increased lactic acid, and then the pH because of this can be decreased. When the pH is decreased, when you see all these reactions occurring, morphologically when you try to look into the cell, there can be a clumping of the nuclear chromatin. Now on the other aspect, because of the decreased ATP, what are the other things that you can see is the detachment of the ribosomes. The ribosomes, they get detached, and as a result of which naturally you see that there can be a decreased protein synthesis. So these are the functional derangements because of ischemia and because the functions of the mitochondria is altered, and as a consequence of which there can be some different morphological changes because of this biochemical reactions. When you look into the mitochondrial damage, which is called as the powerhouse of the cell, so the possible cause of mitochondrial damage would be the increase in the cytosolic calcium, reactive oxygen species, I told you, superoxide anion, hydroxyl ions, and hydrogen peroxide. These are the reactive oxygen species. And then further, there can be oxygen deprivation. And because of all this stress, the mitochondrial genes can have as well mutations. And what could be the consequences of the mitochondrial damage is there can be a failure of the oxidative phosphorylation, which is one of the most important functions of the mitochondria. And next would be the activating apoptotic pathway. When you try to look into this picture, you see that because of decreased oxygen supply, there can be toxins, and there can be some effects due to radiation on the mitochondria. Whenever there is a damage, naturally there is a dysfunction of this particular organelle, that is mitochondria. Whenever there is a dysfunction, you know that the ATP generation is decreased, and as a result of which there can be multiple cellular abnormalities which can lead to a kind of a cell death. Whenever we say cell death, it is in two forms. One is the necrosis, the other one is the apoptosis. So here, whenever decreased ATP generation due to multiple cellular abnormalities, the cell can succumb to a kind of a death state, which is called as necrosis. When you look into the other aspect, what happens is whenever there is decreased survival signals and DNA or protein damage, there can be a pro-apoptotic proteins which can be introduced, as a result of which there can be anti-apoptotic proteins which will have its effect on the mitochondria. Whenever you have such effect on the mitochondria, there can be a leakage of mitochondrial proteins. The ribosomes get detached, as a result of which there is decreased protein synthesis, and ultimately the cell can try to undergo a form of cell death, and this particular form we call it as apoptosis. And this is a different form of a cell death which we call it as a programmed cell death. Now, what happens due to loss of calcium homeostasis? What could be the causes of that? The cause could be the damage to the cell membrane, damage to mitochondria, and damage to endoplasmic reticulum. And along with that, there can be an increased intracellular calcium and opening of the mitochondrial permeability transition pore, due to which there can be an activation of enzymes and induction of apoptosis, what we saw in the previous slide. Now again, when you try to look into this picture, you have an injurious agent which has caused the cell injury, and you see the extracellular calcium is trying to get inside the cell. So increased cytosolic calcium you are having here, as a result of which there can be activation of cellular enzymes. You know, all these enzymes are degradative kind of enzymes which has a ill effect on the cell and the cell membrane. Now, due to activation of cellular enzymes, phospholipase, so there is decreased synthesis of phospholipids, and because of the protease enzymes which is secreted, there can be a disruption of the membrane and cytoskeletal proteins. Due to both of these things, you see that it has got a very damaging effect on the cell membrane, and the membrane gets damaged. Now, due to the activation of these cellular enzymes, you see that there are other two enzymes also which are synthesized. One is endonuclease, which has got a very bad effect on the nuclear membrane, as a result of which the nuclear membrane they get damaged, and as a result of increased calcium, the ATPase enzyme also it gets activated, and when it gets activated, it has got its ill effect on ATP synthesis. The synthesis of ATP is decreased, as a result of which again the functions of the cell and the cellular organelles it is deteriorated. Whenever you see that there is accumulation of oxygen derived free radicals, what we discussed, there can be, they are very important in the cell damage. One is the chemical and radiation injury, ischemia, reperfusion injury, or cellular aging, or microbial killing by phagocytes. Now, these reactive oxygen species, what we discussed, they are a type of oxygen derived free radicals which I told you, superoxide anion, hydroxyl ion, or hydrogen peroxide molecules. So they are produced normally in cells during the mitochondrial respiration and energy generation. They are produced normally in cells during mitochondrial respiration and energy generation. Oxidative stress. There can be a production of the reactive oxygen species increases, or the scavenging systems are ineffective. The result is an excess of these free radicals, leading to a condition called as oxidative stress. So how can these free radicals be removed? And there can be the L effects which can be removed is by the influx of the antioxidants. When we say free radicals, they are also called as oxidants, and to remove these free radicals we always require antioxidants, and this happens by binding of the ions to the storage and transport proteins, and the enzymes which can be involved are the catalase, superoxide dismutases, glutathione peroxidase. Now, what could be the pathologic effects of the free radicals? So among the three, the first one would be the lipid peroxidation in the membranes, the oxidative modification of the proteins, the lesions in the DNA. Now, when you see the defects in the membrane permeability, the causes of the damage could be again the reactive oxygen species, the decreased phospholipid synthesis, increased phospholipid breakdown, and the cytoskeletal abnormalities. The mitochondrial membrane damage could be one of the major consequences. Along with that, we can have plasma membrane damage and injury to lysosomal membrane. And whenever you see there is a damage to DNA or the proteins, what could be the causes? They could be mainly the exposure to the DNA damaging drugs, the radiation, the oxidative stress, the mutations, and the improper protein folding. As a result of which the consequences could be the cell death. And when I say cell death, it means in either of the two forms, one is necrosis or apoptosis. So we'll try to cover the next portion of the cell injury in our next class. And I would like to flip through all the important